So now let's go for the blasting. So for doing the blasting, go to any of the web browsers and search for NCBI and go to the website of NCBI. Now in the interface of the NCBI, we can see option popular resources where we can find an option for this blast. So click in the blast. So this is the interface of the blast where we can find nucleotide blast as well as protein blast. As we need to go for nucleotide blast, click on the nucleotide blast. This is the interface of nucleotide blast. This blast n represent the nucleotide blast, and we can see a box with a enter query sequence, in which we have to enter the accession number of the FASTA sequence. So for this, we have already downloaded a FASTA sequence from NCBI. So copy it and paste it in the enter query section. So either we can go for the accession number sequence and we can choose the title, and we can continue with the default settings which are present in the interface, and finally we can click the blast button. Let me take some time. So this is a result of the nucleotide blast. Here we can see the accession number and the name of the nucleotide sequence. uh, Here we can see the molecule type, query length, etc. And in the descriptions, 100 nucleotide sequences which are having the highest similarity with the query sequence is present. These are the 100 nucleotide sequences. So in this we can see here there is a maximum score. So the maximum score is based on the alignment length, match and mismatch scores, gap analysis, etc. Usually a high maximum score generally means that there is a high degree of similarity between the query and the subject sequence. Here we can see the percentage or the percent. So the percent indicate the percentage of matches between the query sequence and the subject sequence within an aligned region. It is calculated like the number of identical matches divided by length of the alignment into 100. So that's about the description, and going to graphic summary. This is the graphical representation of the similarity between the query and the nucleotide sequences we get in the result. So each line here represents the nucleotide sequences. The blue line is the query sequence, and the red lines over here are the nucleotide sequences that we get from the result. So this graphic summary is actually based on the alignment scores. Here we can see that the lines over here are present, are having red color. So the red color shows a higher similarity with a alignment score of greater than or equal to 200. Pink lines show higher similarity index but less than red with an alignment score of 80 to 200, and green lines show moderate similarity with the alignment score of 50 to 80, and blue lines show less similarity with the alignment score of 40 to 50, and black or gray shows very less similarity with the alignment score of less than 40. So here we can see in these regions the lines are not complete, which means that the nucleated length is not up to the query. Now going to alignment. This is a comparison. In the alignment section, we can see the comparison between the query nucleotides and the subject nucleotides. So this is the comparison with the first result. Downward, this is the comparison of the nucleotide with the second one. So here we can see this is the nucleotide sequence of the query nucleotide from number 1 to 60, and this is the nucleotide sequence of the subject from 1 to 60. So if two nucleotides are connected by a line, it shows that they are same or similar. Here you can see the nucleotide sequence of the query from 61 to 20 and the subject from 61 to 120. So here we can see that there is an absence of line over here, which shows that here there is no similarity between the nucleotides. Here we can see the nucleotide of the query is thymine and that of the subject is cytosine. As they are not same, the line is absent over here. So that is about the alignment. And here we can see the taxonomy. In taxonomy, we can see the lineages taxonomy, and in organisms, we can see the nucleotides of different organisms present in the result. So from the result of the nucleotide blast, we can create the phylogenetic tree also. For that, we have to go to the other reports, and here we can see option of distance tree of results. You need to click in that. It would also take some time. So this is a phylogenetic tree. So the phylogenetic tree is a diagram that represents the evolutionary relationship among different species, organisms, or genes, showing how they are related through the common ancestor. So this point of branching indicate a common ancestor. These are the point of branching. So these point of branches indicate that there is a common ancestor between these two groups. So their branches along with their length and indicate their evolutionary path. We can download this from here. Also, we can download the whole report of the result of the nucleotide blast from here. So that's all about nucleotide blast. Thank you.